High-throughput sequence is the process of determining the order of nucleotide bases along a DNA strand in a massively parallel manner, which is why it is called a high-throughput sequencing. Until now, we have been able to generate four different ways to create our sequencing methods. So they can be broadly classified into amplifying using the mass spectrometry, the in vitro cloning methods, the in vivo cloning methods, and the single molecule method. So we'll cover the major generation of technology, basically the hierarchy in which we have covered these topics. And we can begin first of all with Sanger sequencing, which is the most basic form of sequencing a DNA. It completely uses the in vivo cloning method where the DNA is cloned inside an organism and there is a linear increase in the number of DNA samples, unlike PCR where it is exponential. So over here, you have the presence of a polymerase, DNTP, DDNTPs, buffer, a primer that chops up the template at known locations. And the chance of incorporation of the DDNTP is random, so the fragments of all length are produced. So in this diagram, as you can see, we begin by first of all shearing the DNA using nuclease and exposing this particular fragments into a DNTP and a DDNTP mixture. Over here, DNTP is a normal ATGC molecule, and it is going to be simply polymerized along the DNA fragments. The DDNTP nucleotide contains a fluorophore and it is going to stop the elongation process because it lacks an OH at the 3' pole position. So this way we have a chance incorporation of DDNTP in any location. So what we do is we separate our DNA fragments into four different test tubes of A, T, G, and C respectively, where this A, T, and GC correspond to the DDNTP of A, T, G, and C, so that we are absolutely sure that one test tube contains all the random fragments where the A is present and the T is terminal and the G is terminal and the C is terminal. And this way, when we run the gel electrophoresis in four different wells, we can see the individual nucleotides, that is A, T, G, and C, arranged in their uh, weight, arranged in the order of decreasing weight, like from top to bottom. And this can be further analyzed and the entire sequence can be generated out of that. The second generation sequencing technologies is a high throughput sequence technology in comparison to Sanger. It is going to be cloned in vitro, like all the DNA fragments that we are interested in will be cloned in vitro and it will be sequenced by synthesis. So how we begin this process? We start with a method of Solexa technology. It is a way to form the sequence, and it begins by using genomic DNA and attaching it to different adapters. So these adapters are oligonucleotides that can join to both ends of your fragmented genomic DNA, and it can attach to a surface. And at that surface, it bends back in a manner that allows the reverse strand to start forming in the second point of joining. And this way we are able to create a cluster of multiple such DNA fragments. And once this cluster has been created, this is basically part of a foldback PCR process, and this denaturation creates the clusters of the single-stranded DNA molecules, and then the sequencing is performed sequentially using the primers, the DNA polymerase, as well as the the fluorophore labeled DNTPs. So after the incorporation of a nucleotide, the image is captured and the identity of the first base is recorded. The terminators and fluorophores are then removed, and the incorporation, detection, and identification steps are repeated. So the average read length is currently 40 base pairs, but this can probably increase in the future. The library preparation can be done by transposition, which is part of next era technology. The next era approach utilizes the transposon activity in forming the Illumina compatible paired end libraries. In this assay, the DNA is simultaneously fragmented and tagged with a sequencing adapter in a single reaction. The next era transposition is a random event on the template DNA molecules, but it may favor certain sequence regions over the other. So lowering the copy number of input DNA molecules decreases the diversity of the libraries, and hence it increases the, or like it affects the sequencing result. Basically, these transposons or these tags are attached with oligonucleotides that go on the target DNA and it fragments it. So they create random fragments libraries based on the concentration of transposon that are seizing the target DNA. These tags actually help us to differentiate between multiple experiments because you can run different kinds of experiments on the same DNA fragments. So you can help to differentiate them using these tags and say that this particular DNA fragment cluster belongs to a different experiment. With the Illumina sequencing technology, it uses the clonal array formation and a proprietary reversible terminator technology, which basically includes the fluorophore tagged DDNTP, sorry, not DDNTP, a normal NTP, the DNTP, with the fluorophore that can be removed with the use of TCEP enzyme. And this helps the particular stage of analysis because every time the polymerase comes and binds with the new complementary nucleotide, it will not read the previous nucleotide's fluorophore and it will actually, the laser will excite the new DNTP's fluorophore. So in Illumina sequencing technology, the cluster is generated in the following fashion. The sequencing templates are immobilized on the flow cell surface, and it is designed to present the DNA in such a way that it is accessible to the enzymes while ensuring high stability of the surface bound template and low non-specific binding of fluorescently labeled nucleotides. This solid phase amplification creates up to 1000 identical copies of each single template molecule in close proximity. So this is inside a well of 1 micron or even less the diameter. Now the sequencing by synthesis, which happens in this technology, uses 4 fluorescently labeled modified nucleotides to sequence tens and millions of clusters present on the flow cell surface. So these nucleotides are designed with the reversible termination technology. So that allows each cycle of the sequencing reaction to occur simultaneously for all the clusters in the presence of all 4 nucleotides. And in each cycle, the polymerase is able to select the correct base to incorporate, with the natural competition between all 4 alternatives leading to high accuracy. And this is better than the methods that use just one nucleotide at a time. So it is better than Sanger sequencing in this fashion. And the sequences where a particular base is repeated, for example, in homopolymers, they're addressed like any other sequence and resolved with high accuracy. The analysis of this pipeline occurs in a massive quantity of sequence reads in parallel. So the deep sampling of more than 10-fold even coverage is used to generate a consensus and ensure high confidence in determination of the genetic differences. The deep sampling allows the use of weighted majority voting and statistical analysis, similar to the conventional methods to identify the homozygotes and heterozygotes to distinguish the sequencing errors. Each raw read base has an assigned quality score so that the software can apply a weighting factor and generate the confidence score. Now, the steps that are taken to create such a analysis are in the following 12 major steps. So first, we prepare the genomic DNA sample, where we are randomly fragmenting the genomic DNA and ligating the adapters to both ends of the fragment. Then second, we bind the single-stranded fragments randomly to the inside surface of the flow cell channel. Third, we bridge amplify. So we add unlabeled nucleotides and enzyme to initiate a solid phase bridge amplification. Fourth, the fragments become double-stranded. Over here, the enzymes incorporate nucleotides to build double-stranded bridges on the solid phase substrate. Fifth, we denature the double-stranded molecules. So this denaturation leaves the single-stranded templates anchored to the substrate. Sixth, the amplification is completed when the several million dense clusters of double-stranded DNA are generated in each channel of the flow cell. Then we determine the first base. So this is going to happen with the laser excitation. The first sequencing cycle begins by adding 4 labeled reversible terminators, primers, and DNA polymerase. Then we image the first base. So after the laser has excited, the emitted fluorescence from each cluster is captured and the first base is identified. Now, once this has been done, we can determine the second base, and the next cycle repeats the incorporation of 4 labeled reversible terminators, primers, and DNA polymerase. 10th, the image second chemistry cycle. Over here, the laser excitation will cause the image to be captured as before, and the identity of the second base will be recorded. Then we can sequence over multiple chemistry cycles. So the sequencing cycles are repeated to determine the sequence of bases in the fragment, one base at a time. And finally, we align our data together to compare with some reference, and hence the sequence can be determined. Another second generation technology is the 454 sequencing, which is also called pyrosequencing. Over here, the method is highly parallel and it has a two-step approach. So first, the DNA is shared and oligonucleotide adapters are attached. Each fragment is attached to a bead, and the beads are PCR amplified within droplets of an oil-water emulsion. This generates multiple copies of the same DNA sequence on each bead. Second, the beads are captured in picoliter-sized wells in a small fabricated substrate, and the pyrosequence is performed on parallel for each DNA fragment. The nucleotide incorporation is detected by the release of an inorganic pyrophosphate, which is a PPI molecule, and it leads to the enzymatic generation of photons. So the PPI is released and it is converted to ATP, and the luciferase uses the ATP to generate light. The cycle is iteratively repeated for each of the 4 bases, and the average read length has already increased from 110 base pairs to approximately 250 base pairs. A method similar to pyrosequencing is called ion torrent, where it is a non-optical sequencing method. And just like the release of PPI during the polymerase reaction, we have a release of H plus ion as a byproduct of the elongation reaction during the action of polymerase. So this H positive release it results in some unit change in the pH, like exactly 0.02 change in the pH, and it is detected by an integrated complementary metal oxide semiconductor, a CMOS, and an ion sensitive field effect transistor, which is an ISFET device. So after the introduction of a single nucleotide species, the incorporated bases are washed away and the next one is added. And this cycle is repeated multiple times, and the intensity is going to be mapped, and we will be able to generate the entire sequence after the ion torrent procedure is over. Another second generation technology is called the SOLID, that stands for sequencing by oligotide ligation and identification. It is conceptually similar to 454, but the sequencing strategy is radically different. Here, the beads are deposited onto the glass slides, and the sequence is determined by the sequential hybridization and ligation of partially random oligonucleotides with a central determined base that is identified by a specific fluorophore. So after the color is recorded, the ligated oligonucleotide is cleaved and removed, and then this entire process is repeated again. So the reads that are generated are currently 25 base pairs, but they will probably increase to more than 50 base pairs in the future. Now, the third generation technology focuses on determining the oligonucleotide molecule by molecule. So it is also called as a single molecule third generation sequencing approach. Here we can begin by first discussing the SMRT process. The SMRT process is the single molecule real-time DNA sequencing. The principle of this technology is that a single molecule of DNA that is template bound and a DNA polymerase is immobilized to the bottom of a ZMW. ZMW stands for zero mode waveguide, and it is basically a technology that is used to constrict light into a photodetector, and this light is going to be generated by the fluorophore, as we will discuss right now. So the DNA polymerase and this DNA template is immobilized on the bottom of the ZMW, and it is illuminated from below by a laser light. This ZMW nanostructure provides excitation confinement in the particular well, and it enables the detection of the individual nucleotide substrate. So we can see the fluorescence that is going to be released for every nucleotide, and then determine the particular sequence that this DNA template contains. The key advantages of using this technology is that we can sequence ultra long reads that can be from 3 to 4.5 kilobases on average and up to 20,000 bases. We can also sequence our DNA in a very short amount of time, and there is no amplification bias because there is no amplification required. We also have no problem with the variant and modification detection because it can be done in the same run, and it is very cost efficient. The only disadvantages that it has are that it is a expensive instrument, so it needs a high performing computer for data analysis and special algorithms, and it also has a high error rate with raw reads, but these are very random, and we can provide more methods to cross check the output of this data. Finally, we have come to the development of fourth generation technologies, which are non-enzymatic and they are single molecule sequencing. So this is the idea of nanopore sequencing. Here, a nanopore protein is inserted in an electrically resistant membrane, and a potential is applied across the membrane. The buffer that contains the ions travel across this membrane through the nanopore, hence creating a current. So as the DNA passes through the nanopore, the flow of ions changes. This creates a current signature depending on the DNA sequence. And this we can detect and correspondingly determine which nucleotide has just passed through the nanopore. So compared to the other enzymatically driven sequencing chemistries, the nanosequencing requires only a single processing enzyme. In this case, a helicase sits on the sticky end of the DNA strands to unwind the DNA, and upon contact the nanopore protein, this helicase activates and starts to unwind the DNA through the nanopore. More importantly, we can also see the direction in which the DNA is going to move. In 1D, where D stands for direction, only one strand of DNA is read at a time, while in 2D, a hairpin adapter is ligated to the blunt end of the DNA, and this allows the second strand to be read sequentially and makes the fidelity of the base calling process higher. The key difference of nanopore against all other technologies is that the nanopores appear to stream the data as DNA passes through them, hence making the data available for analysis in real time. And this has been used in clinical settings to identify viral strains in as little as 30 minutes from the start of the sequencing run.